Alrighty, gang, here is Dr. Granier with Bio 177, Chapter 3, Chapter 7, which is our Week 2 content. This is a little bit of a little lecture that you can refer back to, especially if you did not make class or remember all of this stuff that we talked about in class the other day. So let's get started. Chapter 3 is sensory evaluation. So looking at how our bodies sense and interact with the environment. And you can see from the objectives, we want to compare reasons for evaluating food products subjectively and objectively. So subjectively is a little bit of everybody brings something to the table that might be a little different, whereas objectively might be its like size, its shape. It's something that might have a quantitative or a numerical value you can uniformly assign. That's the same for everybody to view. For lists, again, physical, psychological, cultural, environmental influences on food likes and dislikes. We'll get to a little bit of that. Explain how taste and aroma combine to give foods their flavor. So it's basically trying to, how does the olfactory and the gustatory system work together to then in your brain integrate into a flavor that you're perceiving. And if you wanted to conduct a taste test panel, how to accurately set that up to best receive data points that are meaningful and useful for you to utilize. So it starts off with sensory evaluation. So I mentioned again in class that for our sensory system, that is part of our nervous system, and we have receptors that fall into one of two categories. We have sensory systems that are considered general sensory systems that are located everywhere in our body. And examples of that are temperature, pain, proprioception, which is pressure, touch. And again, we have that everywhere in our system. So you can feel this or interact with those types of general senses on your fingers, on your toes, on your back, on your belly, in any location. The other side of sensory system is our special senses, and we have our special senses are vision, taste, which is gustation, olfactory, smell, equilibrium, so dynamic and static equilibrium, also sometimes referred to as your vestibular system, and your hearing or auditory. For food purposes, we don't probably need to talk about equilibrium and hearing because they are probably not going to have a huge influence on your system. For sensory evaluation, smell and taste do play a big role in what we perceive then is ultimately food flavors. Visual will make maybe appealing pictures for us to entice us to maybe want to try food or not. For the general senses, temperature will probably come into play. Temperature extremes will turn into pain receptors, but then again, we also do have independent pain receptors and itch receptors. And then proprioceptors, texture receptors, pressure receptors, touch receptors, that's going to give us some information related to, again, food in our mouth, ensuring that maybe we don't bite our tongue, our teeth don't smash into our gums, and that the food we then feel its texture, its unique properties, again, of viscosity or fluid amount, structure, and components related to proprioception. Okay? So, influences on foods. Why do we like or dislike food? So physically, we have body chemistry. We have a number of taste buds that, with, again, age and gender, might affect our ability to detect flavors. So one of the things that will happen is your taste buds will turn over. Those are cells that are going to be exposed to the elements, exposed to the acid of the food, the base of the food, exposed to saliva, exposed to pathogens in food, exposed to all sorts of environmental shearing, tearing, and with age, with time, we will see a turnover of these cells. And as these cells turn over, they could potentially change what we will maybe perceive as likes or dislikes. Hormones will influence many cells. So whether you're female, male, there should probably, that what I guess this is the point to try to take away here is estrogen or testosterone could make us have certain flavors that, or certain taste palating items that we're more likely to want to ingest over others. The number of taste buds, more taste buds would mean more discernment, more ability to interact with food. So maybe that could make us more likely to eat certain foods or that we prefer. But in cases of non-preferred foods, maybe that would help us not want to eat certain foods that have strong taste, strong being they interact with a larger number of taste buds. Okay, so physically, again, it's related to the number of cells in our taste buds, the number of cells in our olfactory sense, the age of those cells, the age of our bodies, so the number of cells we have, the neurological pathways we have set up that can be maybe modulated or influenced by time, by age, and by potentially estrogen or testosterone, so have a gender effect. On the psychological side, there will be certain patterns and certain habits we form which give us a taste bias. So these can be habits or patterns that lead us more likely to want things or less likely to want things. So it can give us a like or a dislike or a positive or negative experience. So if we have a past experience of maybe after eating a certain food, we succumb to foodborne illnesses, sickness, or maybe not even food related, but just ended up getting sick, we might then associate negatively a food that we ate prior to that as being part of that negative experience. Okay. This is also where advertising might come into play, where we maybe prefer certain brands, labels because of the marketing campaign associated with it. And it could then give us a higher propensity or lesser propensity to want to do or utilize those products. All right. For our culture, there are certain beliefs and behaviors that can influence our food choices. Some cultures are vegetarian as a cultural belief. Some are going to not eat pork or beef. So those are choices then that people grow up as part of their religion, part of their cultural background, part of their identity that they then incorporate into their lifestyle and then make food choices based upon that. Environment, again, if we go back to chapter 1, people ate what they could grow, and you grow what is going to be a sustainable and nice yielding crop. Corn kind of came about with the settlers that came to the United States because that's what we grew here, whereas they were more used to wheat and other products, but then corn became a staple in their diet because it was easily, they learned how to grow it and harvest it and make use of it here. All right. I always think of kiwis again. Most of us would probably never eat a kiwi because it grows only in one location, but because maybe our now environment of more easily being able to harvest and transport foods quickly to other surroundings, we now can incorporate kiwis into our diet if we so wish. All right. Now, my kids are, again, just go talk to a child, and appearance of what does it look like, how big is it, the condition, the color of the product is always going to drive children to want to eat or not want to eat certain items. In our USDA regulations, they always tell people to eat a very colorful plate. So we know that color of food comes from a different chemical makeup, and that represents potential different nutrients, different vitamins and minerals as being available in that food. And that's part of well balanced is eat very colorful and a rich color palette. So you get more variety. But again, sometimes we associate colors with certain, again, learned behaviors. Red is stop, red is bad, red is maybe don't do that. So it might be that certain people will avoid eating certain colored foods because of other cultural, other learned behaviors of those colors as being stop, don't go, don't do, or don't eat, or poison or danger. Okay? So when you think about the last objective of this chapter about setting up your test, you do want to be sensitive to if you have certain lighting or certain, again, sensory, visual lighting candles that could enhance or detract from the colors that you're trying to present on the plate, which might be part of your process to make a more desirable plate, or in some cases a less desirable plate. All right, the takeaway I take is if I want my children to maybe eat something, make them eat it in the dark, so that way they can't potentially have a bias form or immediately decline trying something because of how it looks. All right, so let's review about our visual senses and how this works. So the eyeballs, you have two eyeballs. You are considered biocular. In the eyeball, there are three layers. The outermost layer is the sclera and the cornea. The innermost layer is the retina. The middle layer is the choroid. Okay. In the retina, there are your photoreceptor cells. The photoreceptor cells fall into one of two categories. Cone cells are color sensitive cells that respond to the Roy G. Biv spectrum of light. Rod cells are going to respond to light energy, but are not going to give you color information. They are going to give you in shades of black and white. Within your retina, you are going to have interspersed lots of rods and lots of cones. If we didn't have cones in our peripheral or all throughout our retina, we would see color in one area and then gray scale everywhere else. So we know we have cones everywhere in our retina because we see color in our periphery. All right. The main place that in daytime high light conditions, your cornea, your lens of your eye is attempting to bend light rays to converge on a single spot is typically on your retina in a depressed area called the macula, and in the very center of this depressed area of the retina, it's known as the fovea centralis. And in the fovea centralis, we should have a very large number of cone cells. The larger number makes this area the place where our eye is going to have its best visual acuity, meaning you will be able to discern very minor, very infinitely small, maybe pieces or components of information to discern letters, numbers, small text, figures, shapes. Okay, now rods are going to predominantly be everywhere else in the retina. And again, your cones are everywhere in the retina, but tend to cluster in large numbers in the macula and specifically in the fovea. Now, cone cells, because they're day vision and they give you color, they are the best cells, photoreceptor cells, to see during daytime conditions. Rods, with their lower light sensitivity, are best for nighttime conditions. So daytime transitioning to nighttime or night to day, so dawn and dusk, are some interesting time points for our eyeballs because we're beginning to utilize both of these photoreceptors somewhat in tandem. And again, that could potentially have some conflicting information. And again, if you look at data related to car accidents, most car accidents are going to potentially happen in the dawn and dusk hours. And there's a variety of reasons. One of them can potentially be attributed to the mismatch of information between what the rod cells and what the cone cells are independently sending to your occipital lobe of your brain. Some of it's related to the time and the circadian rhythm and the alertness and the awakeness of the body and the time of day and the work conditions and things like that. But for the most part, there is data to show that at dawn and dusk, when you utilize the rods and cones in tandem at the same time, that neither one is doing their best ability because of the conditions to give you visual information, and so they can somewhat overlap and have conflicting information. Okay? Now, at night, you use predominantly your cells because your rod cells are not located in that high number in the fovea, in the macula of your retina. That is part of the reason why if you try to stare at something at night, it may disappear or appear to flicker. And the best way to use your rod cells in a nighttime environment is to employ scanning techniques with your eyes so you are constantly able to manipulate your eye and utilize rod cells throughout the retina. Within the cells, there is a photochemical that is what makes the cell responsive to photons from light energy. In the rod cells, that photochemical is known as rhodopsin, and it comes from the opsin and retinol. And retinol actually comes from vitamin A. So, in relationship to visual acuity and very good night vision, you want to, as a nutritional mentality, make sure you have sufficient vitamin A in your diet, so you have within your rod and in the role of cone cells as well, sufficient retinol to be available to be combined with opsin and form these visual chemical structures that are sensitive to photoenergy. Vitamin A is a fat-soluble vitamin, and again, it comes from our diet mostly. So we don't make it, but once we get it from our diet, we can incorporate it into rods and cone cells, into rhodopsin, and it will be what helps these cells be photosensitive, be light-sensitive special receptors. Okay, so when we look at how do our eyes and how do cone cells and rod cells work, because we have two eyes, each eye is going to have an area of overlap, and with that area of overlap, it's going to allow us to ensure that when our brain takes a visual picture of maybe a 3x3 window, each eyeball is giving information. Some of the information from the right eyeball stays to the right side of the brain, some crosses over. And on the other side, some of the eyeball on the left side is going to stay on the left side and give information to the left brain, occipital lobe. Some will cross over and give to the right occipital lobe. And so with that help and assistance, some of our naturally occurring anatomical and physiological blind spots we do not notice, because as long as the two eyes work together, they can compensate for the lack of information in pieces and parts here and there occurring in the retina. Okay. Now, when the photoreceptors respond to photoenergy, those photoreceptors are going to initiate electrical responses that are going to be carried by neurons from the retina through the optic nerve all the way to the occipital lobe. Again, some of that information has to cross from the right side and the right eyeball to the left side. Some from the left side and the left eyeball needs to cross over to the left brain. The area that crossing over occurs is a very distinctive place where both occipital nerves come together, and some of the fibers or some of the neurons are able to cross midline. That is known as your optic chiasm. Your optic chiasm is very close to your brainstem, your midbrain, these older areas of the brain where we have certain information taken potentially from some of these neurons that are then heading into those regions of the brain to give information for circadian rhythm. So you have an idea of when it's night and day. That in humans can be messed up with artificial light, but in animals, the information of night and day lets them know seasons, lets them know if the days are getting longer or if the days are getting shorter, and much of their behavior of whether or not they go into breeding, they go into heat, is somewhat depending upon that information. So it's very important. If you ever go out to Colorado, all of the elk start to do their mating and their bucking and their fighting over who's the dominant male of and gets to implant and breed with the harem in September. And what's happening in September? Well, that's where the autumn equinox happens, and that's where the days are going to, in a 24-day period, the time that is sunlight versus the time that is dark is going to be more darkness than light. And that's part of how, again, the eyes taking that information of light for 8 hours, but darkness for 16, that's how part of the body of these animals know to move into behaviors of getting ready to prepare for winter and getting ready for breeding purposes. Okay. So the last piece of information is that all of our information is heading to our occipital lobe, which is part of our cerebrum and our human brains. The occipital lobe has these areas known as the primary visual cortex, and that's this place in the back of your head where all of this light energy and all of these electrical impulses that are coding of what your color or what photon energy you're responding to is put and providing you a perception of what you then see as your visual reality. Okay? All right. That's a lot of information, but it's a lot of good information to help you hopefully better understand your visual system. Okay, so let's move away from visual and let's now move into flavor, which can be a combination of olfactory and gustatory. Okay, so flavor is a combination of the two. If we are sick and we can't smell our food, it might then perceive in our brains, again in our temporal lobe where the gustatory and the olfactory senses are heading, that the food is not as flavorful as maybe it really is because we're not using fully both of the senses. Okay, now taste is going to be by chemical receptor cells embedded throughout the tongue and parts of the cheek and the gum and the areas in our mouth. There are 5 basic tastes that we can document as areas of the tongue that tend to have these tastes predominantly there. That's bitter, salty, sour, sweet, and savory. There are other taste receptors related to water, related to heat or spice, related to brothiness. So that's our umami. There are other receptors that potentially are responding to acid base. So that is, again, how foods can sometimes give us some information towards astringency. Okay. Because in our mouth, we do have general senses. We'll have texture receptors, we'll have pain receptors, we have temperature receptors. We can also potentially feel food as it enters our mouth for its temperature of hot or cold. Extreme hot and extreme cold, where we're getting close to those regions where damage can occur physically to cells, that can turn temperature receptors into pain receptors. Chemicals interacting with chemical receptors in the nasal passageways tied to the neurons that will carry information to the olfactory center of our temporal lobe are going to be part of aroma or smell. Okay, so again, for your tongue, the sensation or the special sense of taste is real name is gustation. On your tongue, there are these general areas of rising hills and valleys. Embedded in those rising hills and valleys are a combination of cells that can serve as chemical receptors for taste, mechanical receptors for texture, temperature receptors for temperature and potentially pain. All right. These cells for specifically taste, the chemical receptors, are going to congregate together in these hills and valleys of the tongue known as your papillae. Your papillae are not uniform. You do have 3 different populations depending upon if they're more flat or more pointy. But within those papillae, there will be these regions where the specialized chemical cells are going to push into external environment and have projections capable of interacting with chemicals that are part or liberated from your food that you're eating as you mix that food with saliva and start the process of chemical and mechanical breakdown. Okay, and the combination of those cells along with the underlying neurocells that will take the information from these taste cells as they respond to chemicals and alter their physiological signaling molecules will then lead to electrical energy that can travel through potentially a few of your cranial nerves towards your olfactory towards your gustational center, which is in your temporal lobe of your cerebrum of your brain. Okay, so again, your papillae are not uniform. These are the 3 types. You can see the circumvallate is very more flat, but the filiform is more pointy. Again, the nervous tissue is that center tissue, and you can see that there will be some projections of some of the cells towards the margin, towards the edge of where the papillae physically ends and the external or mouth environment is located. And those projections allow for these cells and their hair-like projections to trap, capture, and pull in molecules that come from food, come from water, come from the food and drink you're eating, and stimulate receptors that can lead to a sensation of taste. Okay, again, for those taste buds, there are basement cells. There are cells that reside there as part of the basement or connection to the underlying tissue. The cells that are going to turn over every 10 or so days that are going to extend cellular projections or hair type projections that allow the cell to then interact, capture molecules from food or drink, are known as your gustatory cells. These are the cells that upon interaction with molecules are going to undergo conformational change. That could mean they release chemicals, so those basal cells either respond and start to trigger electrical impulses or not. All right. Okay, what are your cranial nerves? So your eye was your optical nerve carrying information to the occipital lobe. Most of the tongue is going to be, for the special senses, carry taste information through the facial nerve, which is cranial nerve 7, or glossopharyngeal nerve, which is cranial nerve 9. However, because more general senses are located, there are other senses that play a role in the mouth. So some of the information related to temperature, pain, pressure, viscosity, so consistency of fluid and solidness could be also carried by the facial nerve, the glossopharyngeal nerve, or trigeminal nerve, or the vagus nerve that might innervate other areas of the mouth, the gums, the teeth, and the lips area. All right. We know that caspasian receptors, which are known to be some of our hot or peppery receptors, are going to be carried on the trigeminal nerve. And again, some of that comes from, it might be because those receptors can be found not just on the tongue, but on the lips and on the gums and on the cheeks and on other areas. Okay. All right. Your 4 primary taste sensations: sweet, sour, salty, bitter. Other is unami. Unami receptors, again, were found in Japan, so that's part of the reason why unami for beef brothy and Parmesan cheese gets its name. There are some water receptors that let us, again, have information on the consistency, and they're towards the back of the tongue, and that's twofold. One, it lets us make sure that when we go to swallow food, we're swallowing a more watery, liquidy bolus instead of solid bolus, because again, part of the process of digestion is we need to take our food and physically suspend it as molecules in a watery background material that then can more easily be pulled into our system, which is 70% water. Okay. So again, for odor, which is part of taste and part of understanding and pleasure of food or dislike of food, we have our olfactory sense, which is a olfactory nerve that is going to sit on top of our skull at the ethmoid bone. It's going to have projections that push in through the bone from our skull to our nasal passageways. And the enlargement of the nerve on that ethmoid bone is known as your olfactory bulb. Okay? And the olfactory bulb, much like our taste receptors, is chemical receptor. So it's responding to chemicals that when we inhale air are part of the air environment. Part of the air then has to be moistened, so these molecules are more likely to be trapped in mucus or liquid and then can better interact with the cells that are lining the membrane of our nasal passageways. Okay, so just to give you another idea, cranial nerve number 1 is our olfactory nerve. It is the most anterior. It comes out of the area right here near the hypothalamus, the pituitary gland, and it's going to run on below the frontal lobe, and it's going to enlarge right on the base of the skull, and this bone here is known as your ethmoid bone. And the ethmoid bone has these little air sacs or holes that allow part of these cells that are sitting in this olfactory bulb to be able to push into and become a part of the lining that then is found around and a part of your nasal passageways. Okay, so again, your olfactory organ, which is the material that's pushing here, is a combination of the epithelial or connect the epithelial tissue, the cells that are protecting and forming a barrier of what is air versus what is you. It is going to be part of the specialized cells that push into this epithelial layer to interact with molecules with specialized chemical interactions that lead to us then smelling. And the underlying connective tissue that holds all of these cells to the body and provides a framework for these neurons connected to these olfactory cells, these chemical receptor cells, to then create electrical impulses that can be carried down the olfactory nerve to the temporal lobe and to the olfactory centers where smell is then processed into perception. Okay. So just to give you a better picture, here is our cribriform bone, our cribriform plate of the ethmoid bone, which has holes that allow for, from the olfactory bulb, neurons and cells to push in and be around our protective epithelial cells. These specialized olfactory cells are chemical receptors, and they will produce little hair-like extensions that protrude into the mucus, into the liquidy barrier that is created by our epithelial cells, in an attempt to help us try to trap molecules and particles that are found in your air. So some of what they're trapping could be particles like pollen, could be particles like mold, could be particles like uh, fossil fuel pollutants that we get exposed to when we burn fossil fuels or cars. Okay, so there's a variety of reasons. There are going to be also these glands, which are just additional epithelial cells that are going to create mucus, going to create solution, watery, that can be what you then think of as your snot. Again, to help humidify and to help, again, take some of the airborne particles, take them out of solution, and in doing so cleanse our hair, so the air getting into our trachea, into our lungs, is more pure or less polluted with particles and molecules. And most of those particles and molecules, upon interaction with these olfactory cells, will try to trigger, or will in some way trigger, a potential smell, and that potential smell could then influence what we taste in our food. Okay? For the other piece of taste that is not the special sense, but more of the more general sense, you have receptors throughout the tongue, mouth, and lips that can give you information on chewiness, graininess, brittleness, firmness, the thickness, the viscosity, the water content, so if it's fleshy or if it's solid. And those receptors, again, are going to continuously give you information that helps you determine when to swallow food, so when to use the tongue to initiate a swallowing reflex, which means you want to mostly send a bolus of water-rich mixture of food together with water towards the esophagus and eventually the stomach. You don't want to send something too solid, too large, and still too originally solid, not mashed or mechanically broken, because those things could potentially lodge or obstruct or take longer to get to the stomach, and obstruct, if they fall the wrong way into the trachea, obstruct your airways. Okay. So how to set up a taste panel. Again, there's some rules that you tend to follow. You want to make sure about the lighting, about the colors, about the sample size. You want to make sure that in your any survey sheet. You don't potentially introduce like a value or a letter that people might more readily then associate is supposed to be better because it's the A sample or the first sample. So there's lots of rules that the book goes through on taste test paneling. All right. And again, we can also film and record taste testers so that way you could potentially try to infer what some of their biases might be. And again, you don't want them to see each other because then that other person's response to a food could potentially then influence the other people. Okay. I always think of like the crazy people that want to eat like squid and octopus and like crickets. And, you know, if you walk into a restaurant and everybody's happy and eating it, then maybe you're more likely to try it. But if you walk into the restaurant and people are going, ew, and making funny faces as they eat their food, maybe you're less likely to try it. Okay. So that's just kind of all of that. All right. Again, you want to fill out the evaluation form. There's like a scoring system that the book pushed about less likely, more likely. And then the problem with all of that ends up being you don't get data that's yes or no. You get data that has a grade or might make your statistical analysis more fun if you're into that. But again, hopefully you pay for good statisticians to help you interpret your data. So these are your recaps. That is the slideshow for chapter 3. Where's my slides for chapter 4? There I am. Okay, so let me do chapter 7. Ah, it's blotchy. Okay. So water. Again, water is the universal molecule that provides a background for many organisms to live, breathe, and survive and interact with their environment. Most of the earth is water, so we know that most organisms need to be able to survive in and around water because that was potentially where earliest organisms originated. Okay, now chemically speaking, we need to talk about water is a chemical substance of an oxygen molecule sharing electrons with 2 hydrogen atoms. And so the sharing is unequal, which is going to make water very unique in that the oxygen is going to keep the electrons more so in its shell orbital ring, these are chemistry terms, than the hydrogens would. And so the oxygen, even though by technicality, there are equal number of protons and electrons because the electrons tend to stay in the orbital ring of the oxygen more so than the hydrogen. This is going to be what we end up referring to as a polar molecule. So the oxygen tends to have a more negative polarity and the hydrogens are going to have a more positive polarity. In inorganic chemistry, we learn all sorts of things that water and oxygen do as chemical reaction material makers. They help sometimes break apart molecules and they can help also build molecules. All right. Because again, most of our cells derived from organisms that had to live a watery existence, they need water inside and in some cases outside, and they need to have nutrients move in and out across membranes through a watery background molecule or material. We are 70% water. We can make some water molecules through chemical reactions inside our cells, but the amount of water we make internally is not sufficient for all of our water demands and water losses. So therefore, water is an essential nutrient that we have to ingest with our food to balance ourselves with the water we lose every day, that we are sufficiently replacing it with water gains. Okay. All right. Next slide. So it has 2 hydrogen atoms. By definition, it is supposed to be everything was perfect. It's a covalent bond between 2 hydrogens and oxygen, and it's supposed to be neutral. But because the way the sharing of the electrons is occurring, it is actually a polar bond between the hydrogen and the two hydrogens and the oxygen, and the unequal sharing, so the time that the electrons spend, which tends to be longer in the shell around the oxygen, makes the oxygen area of the molecule have a negative tendency, and the hydrogens have a more positive. Okay, so that's what, again, is shown in this picture. Now, when we start letting hydrogens that bound to other atoms are involved in unequal sharing, they can start to then form what's known as hydrogen bonds. And in chemistry, these are very weak bonds that are easily broken and easily made. But these hydrogen bonds are a way that the positive pole of a hydrogen in a molecule can become attracted and form a lattice work or a network of bonding or weaving amongst the molecules, that it'll be attracted to be near and in a uniform, repeatable pattern near the negative part of a different molecule. So because the oxygen seems to be a little negative and the hydrogens tend to be a little positive, you'll notice that water can make a very uniform and repeatable pattern of the hydrogens congregating close to each other, forming a positive area, the oxygen kind of forming a negative area, and those hydrogens of one molecule become somewhat attracted and line up and orient themselves to be near the negative or the oxygen of a different water molecule. And when we look at that in different states, whether it's a solid state, a liquid state, or a gaseous state, this polarity is part of the reason why water can freeze. And when it freezes, it actually can expand in its density instead of reduce its density. The other thing is it provides a background framework that then we can put solutes, we can put other ionic, other non-ionic, other polar molecules into, and they can be in suspension. So that means you would not maybe physically know or realize they're there. They're in the suspension of the water, and they don't necessarily fall out or separate from the water lattice work or network. Okay. Again, because of the way water is as a polar covalent bond, if I put water on the table, it's going to bead into a little structure, and that beading and that pulling of the hydrogens and the oxygen towards each other from different molecules creates this condition we call surface tension. Okay, so if I didn't have surface tension, water would be more spread out, and it wouldn't clump or try to bead together. We would see, if we look at water in cylinders, we wouldn't see that kind of dip of water at the center when at the very top of the cylinder when we're trying to read it for height or amount. All right. The other thing that this property of how water interacts with other water molecules is part of the reason why, again, it has its certain melting point, boiling point. It can freeze faster than it thaws, and it can, again, form those crystal structures that make ice a less dense molecule than liquid water. And it helps create an environment that potentially can help put other solutes or molecules and suspend them in the water background. Okay, so hydrogen bonding, if we get back into chemistry and we start looking at physiology, it's one of the reasons why your DNA is double helix. It's one of the reasons why certain molecules, certain things are like proteins take on their tertiary or quaternary structure. So these hydrogen bonding is within a molecule. It's their kind of attraction to other molecules that are also polar, that also have these areas of unequal electron or proton sharing. And they can help, again, with a lot of substances to help us produce a water/other substance material that can have a consistency and a uniform type background. Okay, and it's part of water is what we call a solvent, meaning we can suspend within it a variety of solutes. So we can suspend fructose, glucose, we can suspend some starches, we can suspend some proteins. So carbonic anhydrase, we can suspend trypsin, we can suspend pepsinogen, some different proteins. We can suspend insulin, different hormones that are protein nature. Okay, and again, they are staying in the solution and not falling out or becoming solid state in a liquid and separating away from the water because of those polar bonds, the hydrogen bonds created against the polarity of them and their polar areas in their molecular structure, and the polar and the polarity created in water's structure. Okay, now again, because of the way water works, and because of the way our atmosphere is and the way our environment is, water does play a role in what we determine to be atmospheric pressure. Again, most of the time, atmospheric pressure, standard pressure is because of standard temperature, standard humidity. As temperature, humidity changes, the reading for what your barometric pressure is outside in your yard will change. So if you go to your weather app, typically we are considered in St. Louis probably living at sea level. So standard atmospheric pressure for sea level, which is defined as usually 0 feet above above water, under standard atmospheric temperature and humid conditions, the pressure should read that it is 29.92 inches or 760 millimeters of mercury rising in a barometer. All right. Because again, today it's colder outside. Are we potentially, if we look at our weather app, not reading 29.92 inches, but maybe 30.1 inches? So we are in a little bit of a higher pressure situation because of the temperature. Or maybe we're in a higher pressure situation because of the humidity that we are experiencing today. All right. So pressure is, again, a function of the force of all the molecules in our atmosphere. So all the nitrogen, all the oxygen, all the carbon dioxide, all the water, all the argon, all the helium, all the fluoride, all the neon, all of the gases that exist in our atmosphere. They are in constant motion, and they, along with different reasons, are going to interact with each other, and they are going to exert, when they bump into each other, a force. If we take all of the molecules and we weigh them on a scale, all right, the amount of weight that they push on the scale, if we measure that as mercury rising in a glass thermometer or a glass tube, that would then, if you measure at 0 feet sea level, the amount of particles of water, oxygen, nitrogen, helium, carbon dioxide, carbon monoxide, that should push mercury to a height of 29.92 or 760 millimeters of mercury. If we go to higher temperatures, the amount of molecules that are pushing and available and interacting start to decrease. So as we go up in altitude, so if we go to Colorado, we would find that there are less molecules interacting. So now the force exerted on our mercury to rise up a tube of glass is less. And so the pressure would be decreasing. As we go up in altitude, we start to see the water content of the atmosphere is going to change along with all the other molecules, and it's going to decrease. And so what we find is, as we go up in altitude and there are less water molecules, water, because of its chemical properties, because of its concentration, because of its nature, is going to boil at lower temperatures when we're at higher altitude. And when we potentially go to lower altitudes, or even go maybe to really extreme high pressure situations, because we're diving in a submarine or putting ourselves in a hyperbaric chamber and simulating that we're going into the earth at 100 feet underground, water would then potentially, in those high pressure situations, need a higher temperature to force it to boil. Okay, we know this and we account for this with water content of our food and on our recipes. So most of your boxes for brownies, cake, cookies, mix will tell you if you're above 5,000 feet of altitude for your living situation to consider altering conditions to best then ensure water content of the food is cooked sufficiently and appropriately to give the texture, to give um, the full effect of desired cooking. Okay, so this is important for our respiratory system. This is important for our ears, our body. You know, as we go up in altitude, if you had a bunch of beans, you would have more methane gas. You would have more desire to fart. Um, you know, so it can play havoc in other ways on our bodies related to food, but for the most part, we tend to live between 10,000 and sea level conditions. The water content, again, is going to have an influence on pressure. Right now, if today's pressure is higher than normal for normal standard sea level conditions, instead of maybe seeing our water boil at 100 degrees, we might need 102 degrees of Fahrenheit heat to get our water to boil. Okay. All right, so impurities in water. So again, anytime you have water that is going to contain other molecules, those molecules could be ions and atoms, it could be glucose, it could be proteins, it could be particles that are the product of incomplete combustion, so like methane gas, carbon monoxide, carbon dioxide. It could be something we intentionally put in the environment because we artificially made it. So all of those molecules in water to create water not completely pure, not pure or only water entities, can be described as solutes, and our book continues to describe them as impurities. Now, impurities can maybe influence the boiling point, the um, the the properties of water, its usefulness, and could potentially in that change the characteristics of how water can be an effective tool for helping us cook or clean or induce taste or aromas. Okay. Most of the time, the molecules that are suspended in water naturally do so because they have the ability in their chemical structure to form hydrogen bonds, polar bonds, with the hydrogens and the oxygen of water's molecular makeup. Okay. So again, why do I care about water? You are mostly water, and most of the water in your body, which is 70% of you, is water. So if I was to try to suction out every single water molecule, I would suction out 70% of your body weight. Your water in your body is mostly inside your cells. So when I look in our pie chart, most of the water in your body is sitting in cells. It is part of cytoplasm. That water is going to provide the background material for your proteins, your DNA, your RNA, your amino acids, things to reside within so you can build energy molecules or break energy molecules, build molecules for structural support or take away molecules that form structural support. Cells get their water and get their nutrients, their solutes, and remove their waste products by interacting with water outside of them. So while most of the water, over half, is inside your cells, the other 40 or so percent of water is outside cells. And we have that in two big major compartments. One is considered the interstitial lymphatic compartment. The other one is your blood or plasma of your blood compartment. Anytime we want to add or take away water from our bodies, we take away and add by taking away and adding from plasma. So if I want to get more water into my body, I initially put more water in my blood. More water in my blood is going to make that what we call a hypotonic solution, meaning the solutes in there are less than the solutes suspended in the water of our interstitial, and that'll be part of why osmosis occurs and water moves from our plasma into our interstitial space. When I have now the interstitial space gaining water more so than solutes, that will become now the hypotonic solution versus the inside cell water. And again, through osmosis, I will then pull water into my cells. So to make my body have more water, I input more water into the plasma and I let osmosis take place. If I want to get rid of water, I need to remove it from my plasma through my kidneys or through respiratory or through some other mechanisms, digestive and skin. And as the water leaves and the solutes remain, my plasma becomes very hypertonic. So the solution is very salty. It has a high solute to water ratio, and water would move from the interstitial space into the blood. And then as the interstitial space becomes more salty, higher oncotic pressure, higher osmotic pressure versus the inside of the cell, water that now is in the cell will want to leave the cell and move into the interstitial environment to create it an equilibrium of saltiness that's equal. So again, anytime I want to add or take away water, I add or take away by adding or take away through the plasma. And then I let osmosis guide the conditions between the plasma and the interstitial and the intracellular environment. Now, if you want to look at which organs in our body are the most fluid rich, liver, kidneys, blood are pretty big because they are considered reticular tissues. They are considered very fluid rich. The place where we have the least amount of fluid is actually our bone, because most of the outside material of our cells in bone is a solid calcium carbonate, calcium hepatitate. It's the calcium bound to different molecules that creates a very strong lattice work, a very strong background material, but it's in a solid state, so water is not present to the same amount. Okay, again, we need water to make cells suspend their enzymes and do their physiological chemical structures. We use water to distribute our glucose, our fructose, our amino acids. Fat doesn't like to be in water, but we still use water to distribute it. We just have to then take our fat and bind it with lipoproteins, with carrier proteins to get it into the hydrophilic environment. All right. We use water to eliminate waste, so urea, ammonia, excess ions, excess amino acids. We use water to help us regulate our temperature because of water-specific heat. The amount of energy it takes to get 1 liter of water to move 1 degree Celsius up or down is a huge amount of energy, of joules of heat. So by having water be our background material, we don't see big changes in our temperature as water is held constant, or pretty decently holds temperature constant. We also use water to help us lubricating, especially when we look at organs like hearts and lungs and digestive tract that has a lot of mixing, a lot of changes in cell, tissue, and organ level features. Okay. Again, for water in the body, those impurities, another name that we refer to impurities or molecules suspended in the water is solutes. Again, it's the molecules that are maybe inside or outside the cell, and because they have potentially ionic or charge, or they have polarity, they can easily then become polar and bond with water and polar hydrogen bonding mechanisms. They can then stay suspended in that water environment. So the outside water might have certain amounts, the inside cell water might have a different constitution of molecules. How those molecules end up in one over the other preferred environment really comes down to the cell and the cell's membrane selectively pulling certain molecules in and keeping certain molecules out. Again, some of the key solutes that you need to replace as you lose through a variety of mechanisms that are key to your water balance of your body, as well as other physiological processes, are sodium ions, potassium ions, chloride ions, glucose, calcium ions, phosphate molecules, which in solution of water form a negative molecule capable of polar bonding, bicarbonate ions, which again are a molecule, HCO3, negative charge, that again has potential to interact polarity-wise with water. Okay, so the rest of this really and truly probably is more about how you cook or use water that's in food or outside of food to develop the change in chemistry that leads to food then being a more palatable substance for eating, more flavorful substance for eating, more digestible substance for eating. Okay. So I'm going to let you guys read the chapter on that. Again, most of our foods, similar to our bodies, are high in water. That's why we don't always have to drink all of our water to replace the water lost in a day, because many of our foods bring water in. And again, when we look at water as part of structures, as part of food, as part of ourselves, water that's easily separated from the food is considered free water. So you can freeze it and boil it easy. And it helps. This is the water you remove to dry out food. Bound water is going to be water that is part of other structures. So again, this might be a molecule within the cells of that food item where it has H2O as a part of it. So it's water embedded in other material, and that is still water in the molecule, but it's water that's not easily manipulated. Okay, and then a hydrate is any compound loosely bound with water, and if you boil it, you free the water from the molecule. Okay, again, how the water exists inside foods can influence their longevity, their perishability, their spoilage amount, their freezer thaw capability, and usefulness. Okay, water activity, again, you take into how the water supports enzymes, canned foods more perishable than dried. Again, for water, they do a variety of things, and our goal is to stay hydrated. If we lose 2 to 3% of our body water, we are considered dehydrated. So if you were supposed to be 70% out of 100 pounds, 70% of those pounds is supposed to be water, and you are down 2 or 3 pounds of water, so you are 2 or 3% dehydrated, you can potentially start to see cells not able to have their chemical reactions happen. So those cells could start to be stressed and die. And as cells die, tissues die, organs die, and that could turn to death. So dehydration can kill you. On the flip side, if you overwater, you will maybe explode cells. If you explode cells, they die. If exploded cells happen in large enough amounts, then tissues die, organs die, and you can overhydrate. So it's a slippery slope of we need water, but not too much and not too little. Again, for how does water come into the body? We make water every time we exhale out carbon dioxide. Hydrogen, 2 hydrogens and oxygen combine to form water. So you make water every time you exhale and you release dioxide. You make water every time you make ATP in your cells and you take hydrogens along your mitochondrial membrane and bind them to an oxygen molecule and form a water molecule. So there is water made in your body for a variety of chemical reactions, but the water made is not sufficient to compensate for all the water that you lose. So the other 2 ways we bring water into our system is by food, because food can have high free water content, or by drinking it, which again in pure water or soft drinks or other teas, coffees, things that might be filled with impurities or other solutes with it. We lose water because every day you should have bowels that push excess non-digestible food and material towards the exit hole, and you want that material to stay wet so it's more likely to slide out and not impact or block your exit hole. You lose water every day just as a combination of cell death of your skin. That's called insensible perspiration. And as long as you have skin, that's going to happen. The water could be lost more robustly, like in a fire, when you lose higher amounts of skin and blistering occurs. You could also, again, if you do a lot of friction or you make a lot of blisters, again, you might lose that water faster out of your skin as blister content increases. You need to humidify your air that you breathe, 1, so you can smell any molecules in it, but 2, to help cleanse, and then 3, to help later on in the lungs better support carbon dioxide leaving the body and oxygen going from the air into your bloodstream, which is a liquid environment. So every time we breathe, we humidify the air to better later on cleanse the air and produce a gas exchange that easily lets gases move from a gas to a liquid environment. And the most likely way we lose water is every time we let blood go into the kidneys, we take a sample of our plasma, our extracellular non-living blood components. We return 99.9% of that plasma to the body, but if I'm not perfect, I always lose a little bit. So if you were to think of, okay, if I every minute take 1 milliliter of fluid and I return 99.9% of it, that means I am going to give back to my system 99.9 milliliters, but I am going to keep 0.1 milliliter of fluid and put it in my bladder. If every minute 0.1 is accumulating from your bladders, 0.1 after 10 minutes is now what is it? 0.1 times is 1. All right, so 0.1, 0.1, 0.1, 0.1, 0.1, 0.1, 0.1, 0.1, 0.1, 0.1, that equals 1, because they add up to 10, and 10 would be the 1 goes in front of the decimal point. So every 10 minutes, 0.1 becomes 1, so 1 mil. So 60 minutes and an hour, at an hour later, the 0.1s that you've lost are now 6 milliliters. Okay, if you take, there's 24 hours in a day, if every hour you produce 6 milliliters of water loss, that means in a given day, you are going to, 6 times 4 is 24, 6 times 2 is 12, plus 2 is 14. That's 144 milliliters of fluid produced in a day. So again, our system obviously is not as efficient as only producing 144 milliliters a day. It produces about 1.5 liters, which is 1,500 milliliters per day of water loss. In that water loss, that's how we get then rid of urea, uric acid, ammonia, excess sodium, excess potassium, excess chloride, excess amino acids, bilirubin, some excess hemoglobin, and other molecules that we need to. Okay, for our food, again, we know that food has water and we know food has calories. But when we think about getting water from drinking, not all of us drink only pure water day in and day out. And so the point of these other 2 cartoons on the sheet is to try to show you that anytime you start taking water from other sources, you may take water and some fluid retention of that water, but you also are going to take those other nutrients, those other solutes, those other impurities in, and those other impurities and solutes, if they have a caloric to them, could start to add into your caloric intake for your daily calories, and could put you very quickly into positive calorie balance, which could lead to weight gain. All right. And so if you remember, I said, there are people that go and walk on the treadmill for 30 minutes and then drink a little Gatorade bottle. Well, the Gatorade might have 250 calories of sugar and nutrients inside of it. And the half hour they walked on the treadmill, with the intensity, with the weight, with their height, with their metabolism, might be they only burned 150 calories. So they were thirsty, they drank the entire Gatorade, so they gained 250 calories of food, but only expended 150 calories. So they actually are in positive calorie balance at the end of their workout. And if their goal is to lose weight, that is not going to help them achieve their goals. With every time they work out, they actually put themselves in positive calorie balance. Okay, it's getting kind of long-winded. So again, some things that are considered contaminants or impurities or solutes might, from a chemical reaction standpoint, be damaging or dangerous to us. So that is part of the reason why we have fluid water treatment plants. We have potable water. We try to make sure water for sanitation purposes is not near our latrines, our treatment, our sewage water. In human sewage and animal sewage, pathogens exist. If we then, through fecal oral mechanisms of drinking fecal polluted water, we could be reintroducing parasites into our system that could kill us. So cholera, typhoid. I mean, why in the 1800s, people still took their chamber pots and out their window or out their door, emptied it into the street. And then the well, which is collecting water from the ground and the street and the nearby housing, was then collecting this now fecal-rich water and providing it to the people for cooking purposes and drinking purposes. And so they were, in a sense, contaminating themselves constantly with their sanitation or their means to remove their fecal and urine and body waste. So we don't want water to do that. Again, we have very strict rules. One of the things about China, like with crawfish, is sometimes their water that they flood their rice fields with is known maybe to have higher rates of human fecal matter, animal fecal matter. And so part of the reason, like, Louisiana crawfish farmers say buy Louisiana, buy American, is because the Chinese crawfish might be introducing you to animals that have basically lived their lives in contaminated water sources and might have taken in and be a carrier for pathogens or pieces of fecal matter and pollutants that you wouldn't get with Louisiana grown crawfish. Okay. Again, we try not to use, like if you compost, they tell you not to put your dog or human waste in it because, again, that could contaminate and introduce harmful pathogens that could then contaminate your food. Okay. Other contaminants, again, biological, would be anything like a pathogen. So any virus, bacteria, single-celled organism, multi-cell organism, fungus, anything that it has the goal of getting inside you, taking over, and using your resources to propagate and live, and in doing so damages you and can kill you. Chemical contaminants would be, again, certain irons, metals, molecules that in maybe extreme or high amounts could be damaging to us. Okay. And then physical is like the plastic, the containers, the human-made products that we pollute our environment with. Okay? And again, the big thing about plastics is the estrogens that potentially are driving us to have earlier issues of puberty, and then other harmful chemicals that we ingest every time we drink out of plastic or have plastic containers. We're taking in those plastic, and it's causing us to have issues physiologically in our cells. All right, so that's basically the 2 chapters. I am going to sign off. I hope everybody is ready for the test next week. I have sent it to the printers. I will have Scantrons. Prepare for 46 multiple choice questions related to chapter 1, 2, 3, and 7, and then a little short answer essay related to your scientific method, and showing and demonstrating to me you're beginning to think like a scientist and make hypotheses and data sets to collect. Okay, I'm going to call it a day. Have a great day, you guys.